Hey everyone, welcome to the channel. So in this video we'll be studying about the morphology of bacterial cell, a chapter on the microbiology. So firstly we'll be looking through the shape and size of the bacteria. So this bacteria can be classified based on the shape into three basic groups. So there are three important groups which are cocci, bacilli, spirochetes. Cocci are round shaped, bacilli are rod shaped, and spirochetes are spiral shaped. So these are the basic forms, but there are other types or other forms of shapes also, like Coccobacillus, fusiform bacillus, vibrio, which is comma shaped, and spiralum. So these are the different shapes that can be observed. So there is one more type of one more classification of shape, which is pleomorphic, that is many shaped. This the arrangement of bacteria is also being an important factor. So in cocci, the arrangement of bacteria, if it is paired, it is called diplococci. If it is in the form of chains, there is an example which is streptococci, and for grape-like clusters, it is staphylococci. So the range or the size of the bacteria is 0.2 to 5 micron. Moving to the structure of bacteria, the structure can be studied in five main subheadings, which is comprising of cell wall, cytoplasmic membrane, cytoplasm, structures outside the cell, and the bacterial spores. So you can see the picture that I've put. There is a bacteria, and it shows the longitudinal section of it. Okay, you can see a capsule around the bacteria. There is cell wall also, plasma membrane, cytoplasm, ribosomes, plasmids, villi, and bacterial flagellum, a nucleoid which is with circular DNA. So that covers the structure of bacteria. So firstly, we'll go through cell wall in this video. So what is cell wall? A cell wall is the outermost component which is common to all kinds of bacteria except mycoplasm, because these species are bound by cell membrane and not cell wall. Moving to the next part, the next point, which shows that the external cell wall consists of a capsule, flagella, and pili, which are less common components, and cell wall is located external to cytoplasmic membrane and is composed of peptidoglycan. So this peptidoglycan gives a structural support and maintains the shape of the cell. Okay, next part, which is cell wall of how gram positive and gram negative bacteria are. So I've just put the picture which shows a gram positive cell wall and a gram negative cell wall. You can observe that the gram positive has a plasma membrane, a periplasmic space, and a peptidoglycan to the outermost layer. But in gram negative, there is a plasma membrane to inside to outside, I'm telling it. So from plasma membrane, there is periplasmic space between peptidoglycan and not between either sides of peptidoglycan, and there's an outer membrane of polysaccharide and protein. So gram negative is composed of many layers, so it is very difficult to stain a gram negative bacteria. The structure and chemical composition and thickness of cell wall differs for gram positive as well as gram negative. So the peptidoglycan layer is thicker in gram positive than in the gram negative bacteria. You can observe that there is a purple color line in gram negative, right? That is a peptidoglycan, and in the red thing that is seen around the gram positive, the outermost layer is the peptidoglycan. So there is a difference in thickness of peptidoglycan. So this gram negative has a complex outer layer which consists of lipopolysaccharides, lipoproteins, and phospholipids. And between these outer membrane and the cytoplasmic membrane, there is a periplasmic space, which is a site in some species. So this site consists of beta lactamases which degrade penicillin and other beta lactam drugs, making the bacteria resistant to penicillin. Okay. Then the properties of cell wall are the gram positive bacteria contain endotoxin and lipopolysaccharides. Uh, Its polysaccharides and proteins are antigens that are useful in laboratory identification. That is, the polysaccharide and protein that is present in cell wall help in detection of the bacteria. Then its porins play important role in facilitating any smaller hydrophilic molecules into the cell, thereby giving nutrition like sugars, amino acids, vitamins, metals, and antimicrobial drugs like such as penicillin also move through the porins. So that is a property of cell wall. Moving to the cell wall of an acid fast bacteria. Note that gram positive, gram negative, and acid fast bacteria are different. So a classic example of acid fast bacteria is Mycobacterium. So Mycobacterium tuberculosis is always referred to as acid fast bacteria. So these bacteria have a very unusual cell wall which do not absorb the gram stain, and this makes the bacteria an classic example of acid fast bacteria. Acid fast. So the bacteria is said acid fast because they are resistant to decoloration from the acid alcohol after being stained with carbylphistin. So this property is related to the high concentrations of lipid called as mycolic acid that's present in the mycobacterium. So this mycolic acid prevents the staining of gram stain, and is very difficult to observe under gram stain. So this is the main property that highlights in an acid fast bacteria. So this peptidoglycan, a component of cell wall, is a complex interwoven network that surrounds the entire cell and is composed of a single covalently linked macromolecule. It is found only in bacterial cells. This peptidoglycan component is seen in bacterial cells only, and it acts as a rigid support to the cell, thereby keeping the shape of the cell. And peptidoglycan can also be called as murin or mucopeptide. Lysosomes are also present. These lysosomes go and degrade the peptidoglycan and cause the breakage of the peptidoglycan. So that covers peptidoglycan. Moving to the lipopolysaccharides. So lipopolysaccharides are usually found in the gram negative bacteria endotoxin. So note that lipopolysaccharides are found in the cell wall of gram negative bacteria. It is responsible for many features, like they contribute in acquiring fever and shock in human. So shock usually is because of hypotension. There is low BP because of the bacterial infection. Moving to the next type, which is teichoic acid. These are fibers which are located to the outer layer of the gram positive cell wall, and they're composed of polymers with either glycerol phosphate or ribitol phosphate. So the medical importance of teichoic acid is it induces inflammation and septic shock. So inflammation is allergic reaction. So it is caused by certain gram positive bacteria. So that covers cell wall. So in the next video, I'll be covering the cytoplasmic membrane. I hope you like the video. Just share or subscribe to the channel to get the updates of the videos that I'm going to be uploading in few more days. Hope you like the video. Thank you.